WSU, and the title of her presentation will be Determining Cold Hardiness in Washington Blueberry.
And just a note, if anyone had questions that didn't get addressed, you're welcome to talk to the doctors after the presentations.
Thank you. All right. Can you hear me? All right. Okay. So this project, Determining Cold Hardiness in Washington Blueberry, is a project that I'm involved with. I'd like to acknowledge the other investigators before we begin. That is Gwen Hoheisel. She is in Prosser County as an extension educator, and she is the lead PI for this particular project and plays an important part in terms of looking at cold hardiness in eastern Washington Blueberry. And she works closely with Melba Salzar, who is with the Cold Hardness Lab and helps us with the modeling and some of the statistics that we'll be doing in the future for this project. And then as for myself, I play the Western Washington counterpart. So we are looking at Western Washington blueberry cold hardiness, as well as how these plants acclimate and deacclimate over the course of the growing season. So we began this project last year. We are generating first year data for both parts of the state. And so I'm really excited to introduce and talk more about this particular project. And I'm also really excited for future data that will come out because we'll be able to look at similar varieties in different parts of the state and how they are accumulating cold hardiness, how hardy they are at various temperatures, and how they deacclimate, which will be really important in terms of how we think about managing and protecting from winter or cold injury. So the general outline will be to first give a general review of some of the mechanisms of freezing injury and how plants avoid it. And then we'll go into a few strategies that growers can implement to try to minimize the risk of injury due to cold temperatures. I'll review a little bit of acclimation and deacclimation and talk about physiologically why this is occurring and how this is occurring. And then I'll go into and discuss more about the Cold Hardiness Project and some of the results that we have to date. So number one question is, well, how does cold hardiness occur? And low temperature is, or how does cold injury occur? And low temperature, it's important to remember, is one of the main limiting factors that determine plant distribution. And freezing injury, which is different than other types of injury due to temperature, is damage due to the plant tissue due to temperatures at or below the freezing point. Chilling is different. Chilling is above the freezing point, and it's really something we're not too worried about from an injury perspective. And then freezing can be both extracellular, which means it's outside the cell, or intracellular, which means it's inside the cell. And I'll go into this a little bit more so that we can understand this, because my firm belief is that understanding the physiology and the mechanisms gives us a better understanding to predict hardiness in our own field and implement management strategies and think about this more critically. So extracellular injury, this is, again, the injury that occurs from freezing outside of the plant cell, and it is the most important that we are concerned about for perennial plants like blueberry and other woody perennials. Now, this freezing doesn't directly kill the cell or the plant tissue, but it's actually the formation of ice crystals outside the cell that have a dehydrating effect on the cell and can kill it. This is an image from a blueberry bud collected here in Western Washington that we froze down to minus 28 degrees Celsius. And you can see that the floral initials are damaged as a result of the cold temperatures. And we surmise that this is due to extracellular injury. So let's think about this a little bit more closely. So extracellular freezing, as I mentioned, is freezing that occurs outside of the cell. And here's my attempt at drawing a cell in PowerPoint. So we have the extracellular cell space that is highlighted, that's again outside the cell. And then we have the cell itself. And the cells are over 90% water. They're essentially liquid with a few other solutes and organelles that help the cell function. And ice is dry. So when ice forms outside of the cell, water moves according to its physical water gradient. We have liquid water from inside the cell where it's at high concentration, move down its concentration gradient to that dry ice. And as a result of that, we get dehydration of the cell. And that can cause some pretty severe problems if the plant is not adapted to that type of stress. Now, intracellular freezing, again, this is freezing that happens inside the plant cell. This rarely occurs in nature, but it's seen in laboratory experiments. It happens when we have very rapid freezing. And when ice crystals form inside the cell, we get direct damage to the cell internal organs and those membranes, and we see some non-recoverable injury. So plants have developed mechanisms to avoid freezing injury, and also they have mechanisms to tolerate freezing injury. So plants tolerate freezing injuries by doing various physiological things. They can change their membrane composition because when we have dehydration, a lot of times it's the cell membrane that gets damaged and can result in reduced functionality and death. So one thing that plants do is they can increase the solute or the concentration in their cells of substances. And what this effectively does is it lowers the freezing point. I'm from the Midwest, and when it gets cold, we put ice on our roads. And this is very similar. We put solutes on that ice and we lower the freezing point so that the chance of it freezing again will occur at lower temperatures. Other plants, they avoid freezing injury. They also increase the solute content because if we increase the solute contents, we reduce the temperature at which freezing occurs inside the cell. Some plants can supercool certain cells or certain tissues, and what this means is, aside of having a really interesting name, supercooling, and the plants themselves and physiology is super cool. Am I not right? What this does is that the plants are able to keep water in a liquid state down to minus 40 or minus 40 degrees Celsius. And this has been seen in buds and in certain tissues of certain plants. One thing that plants are also able to do to avoid injury due to cold and freezing is they can have a low growth habit. Half-high blueberries that were grown in the Midwest that are a mix of high bush blueberry and low bush blueberry are a good example of this. That snow cover can provide an insulation effect from colder temperatures in the ambient outside air. So now let's talk a bit about acclimation and deacclimation. So acclimation is also known as hardening off, and this is just the general process by which a plant accumulates hardiness to cold temperatures. And it involves changes in the plant's physiology. It is set off by different environmental cues, such as changes in photoperiod or the amount of light and dark in a 24-hour period. Day length, as I mentioned, day temperatures as well. As temperatures decrease from summer into fall into winter, that's a signal to tell the plant it's time to start acclimating or hardening. And even some drought can induce hardening off. But I wouldn't go in so far if you're interested in trying to encourage hardening to deprive your plants of water. Deacclimation is different. This is the process that the plant comes out or rehardens is sometimes what it's referred to. And this is the process where the plant comes out of its winter dormancy after its chilling requirements have been met. And this occurs in late winter, early spring, and cold hardiness as a result of this process decreases. And it continually decreases as the plant grows, as we go more and more into spring conditions. And this is the way the plant is getting ready to resume growth in the spring and in the summer after being dormant during the winter. And then some plants can reacclimate. So winters and springs aren't always decrease in temperature and increase in temperature. We get fluctuations, and some plants are able to acclimate, deacclimate, and reacclimate and increase their hardiness again afterwards. So this is an example from cherry. It's covered up, but this is a figure that's courtesy of Melba Salazar, and it illustrates the acclimation and deacclimation process. So we have temperature on the y-axis, and we have time and months on the x-axis. Is that right? And what we can see is the plant is acclimating or hardening off as we go from November through January. So it's more tolerant. It's able to survive colder temperatures as it acclimates, and then it deacclimates from January moving forward. And it's different for different varieties. And here we're looking at the variety Chilan, Bing, and Sweetheart. When we surmise that blueberry is probably similar, we see differences in northern high bush and southern high bush, and we would expect different cold hardiness in different cultivars of high bush blueberry. Now, chilling requirements, I mentioned that. So perennial plants require a period of cool temperatures to complete their development and to resume normal growth. If we don't have sufficient chilling met for the plants, we tend to see abnormal patterns of growth. So chilling is just the period between 32 degrees Fahrenheit and 45 degrees Fahrenheit, and the plant is somehow able to monitor the accumulation of chilling hours. Once those chilling hours have been met, that requirement has been met, the plant is now prepared to come out of that dormant stage and resume growth. Now, chilling varies according to types of blueberry. For northern highbush blueberry, which we grow here, it can range anywhere from 800 to 1500 chilling hours. For southern highbush blueberry, it is less, 200 to 600 hours, and the rabbit eyes, it's somewhere between 300 to 600 hours. So what are some factors that influence freezing injury? Well, there's a lot of variables that to consider. One is how cold it gets, the rate of the temperature decrease. The faster the temperature drops, the less prepared a plant is to withstand that cold temperature assault. The duration of low temperatures, or how long those low temperatures persist, can also affect. The rate of thawing can influence injury as well. A gradual thaw, the plant and its cells are able to adjust to the different changes and stresses that freezing can bring, as opposed to a rapid thaw we see more injury as a result of a rapid thaw. And then the physiology and the genetics of the plant can all interact with this. So it's complex. So what can be done to prevent freezing injury? Well, one is to encourage plants to go dormant and to stay dormant. One thing you can do in relation to this is selecting adapted varieties and or species and selecting adapted cultivars. So a good example here in Western Washington and in British Columbia is Legacy. Legacy has some southern highbush germplasm in it, and it is not adapted to cold temperatures in this region. We've been seeing issues due to that. So when we select our cultivars, we need to be mindful of the hardiness and also the time that that variety starts, the chilling requirements, and that time that variety or that cultivar will start to bloom. If there's early bloom times, we are more likely to see injury due to cold because we have colder temperatures earlier in the spring as opposed to later in the spring. Another thing that you can do is to avoid late season fertilizer applications, primarily nitrogen is what I'm talking about, because if we're applying late season applications of nitrogen and we're encouraging plant growth, the plant is all of a sudden switched into a different stage. The stage it should be in is preparing for winter and cold winter temperatures. But if we're applying nitrogen, the plant is all of a sudden in this mode of growing, and we can produce tender tissue that is more sensitive to cold and winter injury. There's a few other things that we can do from the management perspective. One is site selection, so avoiding low spots in the landscape where cold air can sink and settle. Heaters, propane heaters specifically, are something that's also an option, and I've seen these practiced in Eastern Washington, as well as wind machines. And wind machines, what they do on cold, clear nights when we have these radiation freezes, they will be turned on and they will break up the layer of cold air and warm air on top, and they can elevate the cold temperatures to a warmer temperature and reduce the potential for winter or cold injury. Irrigation is another option. I've seen less of this here in Washington state, but it is practiced for blueberry on the East Coast, where irrigation water is applied on the plants because as liquid water freezes and turns to a solid, it releases heat. And keeping that liquid water in a constant state of or turning into a solid keeps that plant and those plant tissues from going below 32 degrees Fahrenheit. Windbreaks also can be helpful in preventing cold drying winds from causing further drying and desiccation during cold temperatures. Timing of pruning is another one that I know growers here are tight on labor, but it's really best to prune when the plants are fully dormant. If we're pruning earlier in that, we can be invigorating the plant and causing the plant to grow. And again, we don't want our plants to grow during that time of the year. In the fall, we really want the plants to go dormant and stay dormant and not produce some of this tender tissue that can be susceptible to cold winter temperatures. There's also other options like protected culture, where we can grow in high tunnels or hoop houses. that can reduce the exposure to cold winter temperatures. And then there's also options out there like surfactants and antitranspirants. These have come out on the marketplace with the idea of, if cold injury is due to desiccation due to the cell, well, let's put on products that reduce desiccation with these antitranspirant products. It's really it's not practiced widely commercially, and there's really little research to demonstrate a benefit of applying some of these products, primarily in blueberry. So these are some images. This is from North Carolina, where we're looking at sprinkler application or sprinkler irrigation on high bush blueberry. Again, as we keep that liquid water in a constant state of turning into a solid, we keep the plants at a temperature of 32 degrees and prevent it from getting any lower than that. And then here's an image from Eastern Washington. This is a commercial field out in the Prosser area. And what I've highlighted here are the propane heaters that are put around the planting and the wind machines. And at certain temperatures, what is done is that the wind machines are activated and the propane heaters are turned on to help elevate the temperatures of that particular planting to prevent cold winter injury. Now, cold hardiness in blueberry, it's interesting. So cold hardiness varies. It varies by variety, which we would expect among perennial woody plants. In blueberry, they can be hardy up to, it says 25 degrees Celsius, but we've been looking at this a little bit in the lab, and it can go up to minus 29 degrees Celsius when they're fully dormant. They can be hardy to pretty cold winter temperatures. But the main factor here is the plant needs to be sufficiently dormant, and that's when the maximum hardiness is achieved. So northern highbush blueberry is the most cold hardy, followed by southern highbush blueberry and rabbit eyes. And the hardiness really varies in the plants. And you can see with this image that I have here on the bottom, what I've shown is that the wood is the most hardiest of the tissue, followed by the vegetative buds, then the flower buds, and then the open flower buds. Really, depending on the stage of the open flower buds, they're only hardy to about 28 degrees Fahrenheit. And the roots also, there's been little work done on cold hardiness and blueberry roots, but what our current understanding is that they really don't attain a lot of cold hardiness. So now I'm going to switch over and talk a bit about the project that I've been working on with Gwen Hoheisel and Melba Salazar. So there's several objectives with this project. The short-term objectives are to first determine an appropriate and reliable method to assess cold hardiness in blueberries. And there's a variety of ways to look at this. So this is a very important first step that we've taken to determine what's the most appropriate method for us so that we can generate good quality, valuable data. Our next goals, and these are developing right now as we're generating the data, is to develop cold hardiness curves for two important commercial varieties, all the way from dormancy to bloom. And then what we would like to do is to update some of the blueberry hardiness charts for key phenological stages. So right now, what a lot of blueberry growers are using to assess cold hardiness for different phenological stages comes out of Michigan State University. What we want to do is develop cold hardiness phenological charts for blueberry in Washington, because Michigan is very different from Washington, and Western Washington is also very different from Eastern Washington. Long term, we would love to develop an automated system where we could measure cold hardiness and evaluate buds so we can generate more real-time data based on the time of the year and the hardiness on the plants. And then we would like to develop a model that we can have on AgWeatherNet so that we can look and anyone can remotely access hardiness information. And this is similar to what we have for grape and other crops. So in 2014 and 2014, Ms. Hoheisel and we looked at several different methods. We looked at the glycol bath method, and this is the standard method for measuring cold hardiness. What you do is you utilize glycol, and this investigator, we were trained at a cold hardiness lab in Maryland just to make sure that our protocols and our methodologies were solid, is you put samples of blueberry tissue in these test tubes, and you put them in a glycol bath, and you gradually lower the temperature. The idea is that you're simulating a slow freeze, and we would bring down the temperatures down to, in my lab, we brought it all the way down to minus 28. On the bottom says DTA, that's differential thermal analysis. So this is a method useful in some types of crops. Basically, you attach probes that monitor as you lower the temperature when there is a release of heat, or we call it an exotherm. And when there's that release of heat or that exotherm, that tells us that the liquid in that tissue, the liquid in the cell, has turned into solid ice. And so there we can estimate that is the temperature where we see winter injury. And then the final one that's on the top right, this is the vending machine method. So here we have our plant tissue samples in this large refrigerator-like device. And we do the same thing that we do with the glycol bath. We just gradually lower the temperature. This is a nice technique, though, because instead of having someone manually remove samples at a given temperature, the machine automatically will kick out a can, and we have our samples that have been removed from that cold temperature and is now starting the gradual thawing process. And it does it all automatically. There's no need to manually be able to pull everything out. And what we found through these trials is that the vending machine works just as well as the glycol bath. So moving forward, what we're doing is that we are looking at using the vending machine in Prosser. So that's what Ms. Hoheisel is using to generate the data. And I'm using the glycol bath method because I don't have my own vending machine. So we just have to stay up late and remove samples, which is not that bad. It's pretty fun. So our approach, what we're doing is we are assessing hardiness once per week in Prosser, and we are doing it once every 2 weeks in Mount Vernon. And the cultivars that we are using are Duke and Draper. And our general procedure, we use different instruments. I mentioned I'm already using the glycol bath. My glycol bath is pictured on this slide. And Gwen is using, or Ms. Hoheisel is using the vending machine. We collect our samples early in the morning. We do our slow, gradual freeze. And then after we've taken out those samples at their designated temperatures, we then undergo a slow, gradual thaw. And then by 3, we are dissecting the floral buds from those samples and looking for injury. And then from that, we can determine at what temperatures do we see a percentage of injury. So these are what some of the buds look like. What we have on the furthest left here is a healthy bud. This is essentially the data that we're collecting after we lower the temperatures. It's green, there's no brown tissue, there's no water soaking. It's viable. It has survived the cold temperatures that we exposed it to. On the middle, I put a question mark there. We have a damaged bud, and there's the vascular connection, which is at the base of the bud, is brown. So in some instances, the conventional wisdom is that that tissue would be dead, or it might not function as efficiently. So when the flowers bloom from that particular bud and the fruits start growing, the tissue is damaged so much that it is unable to meet the demands of that developing fruit. So you might later on have the collapse of those fruits or that flower cluster early on, depending on the severity. But there have been some growers that do their own cold hardiness assessments and say, they've seen the plants recover from this type of damage pretty reasonably well. So this is something we're continuing to look at because, it's not cut and dry in terms of, is this really a damaged bud or not? But then lastly, on the furthest right, this was the image I showed earlier. This is a bud that was taken down to minus 28 degrees Celsius in October. And at that temperature, all of the samples were completely killed as a result of that temperature exposure. So our preliminary results, the study is really ongoing, and the data that we generate is pretty complex to analyze statistically, but I can provide some recent results. So here in Mount Vernon, we lowered temperatures down, on November 9th when we did the assay, we saw 43% bud death at minus 20 degrees Celsius, so that's minus 4 degrees Fahrenheit. And then we saw 100% bud death at minus 28 or minus 18 degrees Fahrenheit. When we ran the assay again on the 16th of November, we saw 20% bud death at minus 4 degrees Fahrenheit. And in Duke, and then in Draper, we saw 40% death at temperatures of minus 19 and minus 20 degrees Celsius, or minus 2.2 to 4 degrees Fahrenheit. And what we can see here from just these small pieces of data is if you look at Duke on the 9th of November and you look at Duke on the 16th, you can see that we're seeing less injury at the minus 4 degrees Fahrenheit. We went from 43% Fahrenheit to 43% to 20%. So the plants are continuing to hardy as time goes on. And our most recent test was on Monday. And so my technician right now is in the lab dissecting buds, and we'll have some data here soon about at what temperatures did we see, what level of death. And Gwen's test will be, next test, well, was on the 2nd of December, so yesterday. And I'm going to bring up this slide again because, again, I'm really excited for some of the data that will be coming out of this project and what we can do with it. And our goal is to, again, develop these types of hardiness curves that can be accessible remotely, hopefully, so that growers will be able to know where hardiness is in their field. And this will also be really important for growers that are implementing cold protection measures, because they will have more robust data in terms of what are the temperatures that they will start to see injury, because then they can be more precise and when they implement some of these cold protective measures that can be pretty costly. And moving forward, again, this is some of the data looking that we'd like to develop. This has been already developed for cherry, and we'd like to do this for blueberry, but we need to generate the data before we can do this. So I'd like to acknowledge several people. We have been collecting these samples from grower cooperators. So Olson Brothers, such as Keith Olson, has been very helpful in this project. On the western side, we've been working with Sakuma Brothers, so I'd like to thank Richard Sakuma. And then we have a few other people, Allison Leonhard, Sean Watkinson, Jeannie Rowland, and Elizabeth Ogden. They run the cold hardiness lab at the Beltsville USDA facilities at Marysland. Marcus Keller and Lynn Mills that have worked with Grape for developing cold hardiness. And then lastly, the Washington Blueberry Commission, which provided funding for this project. And so with that, I'd like to thank you for your attention. Thanks again to our grower cooperators. It really means a lot to have your support and your cooperation to help us get this research done and answer these critical questions. And then I'd like to point out that this picture, I was a little bit of a surprise for me. So I took this picture from a field in Burlington on Monday, and there was a good layer of ice on these plants. And I almost thought it never got cold enough to form ice out here in Western Washington. I'm originally from the Midwest, so it was nice to see something that resembled snow close to me as opposed to not on the mountains. So thank you.
We've got some time for questions here. Sorry, we have some time for questions, and I'm going to start one off with one from Prosser.
Great.
That I think I understand it. The question is whether when one bud is dead, does that mean that one flower is dead? Is it a 1-to-1 ratio, bud to flower?
Sure. So, and I kind of distilled and simplified the data. So when I say one bud is dead, I'm looking at 100% mortality of all the flower initials within that bud. When we do our evaluations, we look at 25% bud death. So, 25% of those flower initials or those little tiny florets are killed, 50%, 75%. So we're collecting more than just dead or alive data.
In your cold temperature evaluation, do you take in the wind chill and humidity and that type of thing?
Sure. Yeah, the question. So, do we take into account wind chill and humidity? And, at this stage, no, we don't do this. These are controlled laboratory freezing assays, and we simply collect the plants in the field, and they're at the physiological stage that we can determine hardiness, and we just do a controlled drop in temperature. So, it just tells us from a temperature perspective how hardy they are at, but it doesn't take into account humidity or wind chill. Mm-hmm.
How cold do you expect it will get on the east side versus the west side? Are you going cold enough? Or, I mean, minus 28 seems much colder than it would ever get here on the west.
Yes, yeah. So, and that's something as we're developing the methodologies, we do have the ability to take it down colder. Documented for high bush blueberry in the literature, they are hardy to minus 29 degrees centigrade. But if, Ms. Hoheisel and Melba need to go lower, they can. Mm-hmm. Oh, will it ever get that cold here as in the east? After living here for less than 2 years and looking at some of the weather data, I would say that's highly unlikely, just given some of the climatic conditions and factors that influence our climate. So we should consistently see Eastern Washington getting colder sooner and getting to lower temperatures than what we would experience here in Western Washington.
Speaking of cold weather, we've just came through a 2-week cycle on the east side where the temperatures range from 8 to 22 degrees as a
Yeah.
high, 8 as a low, which is, I've lived there for a long, long time. That's exceptionally unusual for it to be that cold that early. And obviously, as you have alluded to here, the temperatures, as they get more dormant, they become less susceptible. My question to you, and I love the fact that AWN and potentially the site might be available. Could you expound a little bit more on how often you're going to have that site and that information available, and what time of the year are you going to start giving the fact what just transpired?
Sure. So let me just double check and make sure I understand. So you're asking when will we have this available? Oh, that's a good question, and it won't be for a few years out there. We really have to develop the hardiness measurements and have a better understanding of how cold hardiness, how hardy these plants get within a variety.
Well, I'm just going to expound on that a little bit more.
Sure.
I guess, and because we use freeze chambers pretty extensively on the east side, and most everybody does, you have to. With that being said, in the kind of conditions that we just came through, will you be analyzing the bud or the hardiness of the plants based on the percentage of dormancy they are at that particular point in time? So these are very accurate.
Sure. Let's see. We'll be. Sorry. Can you repeat the question again? Sorry.
Well, I guess what I'm saying is, typically most of us over there, we cut
Mhm.
our fertilizer off early.
Sure.
And so we try to push them into dormancy, and the research has shown that you don't want a lot of shoot growth later on going into the fall. On a year such as this year, and the reason I'm bringing this up is we do use heaters, we do use wind machines to help suppress, frost freeze damage. And on a year like this year, I guess maybe this is a suggestion as much as a question, that there be some efforts made to make sure that as we come into these predicted cold climatic conditions that we just went through, that perhaps they'll look at the hardiness of the plant
Sure.
well in advance to make sure the dormancy is similar to what you just talked about, going from the two different levels of mortality.
Sure. Okay. Thank you.
Okay.
Great.
Thank you, Dr. DeVetter.
Thank you.